Hello students, I am teacher Svetlana. In our today's lecture, we will learn about DNA fingerprinting and applications of DNA fingerprinting, which is covered in the chapter Molecular Basis of Inheritance. So let's get started. DNA fingerprinting. Students, genes present on chromosomes are responsible for determining characters of the organism as well as for the inheritance of characters. Due to recombination of parental and maternal genes, we differ from our parents. Differences also arise due to infrequent mutations that occur during gamete formation, that is during cell division. Now, due to all these factors, every individual has its unique genetic makeup, which may be called its fingerprint. The technique developed to identify a person with the help of DNA restriction analysis is known as DNA profiling or DNA fingerprinting. The technique of fingerprinting was first given by British geneticist Dr. Alex Jeffrey in 1984. Students, DNA fingerprinting technique is based on identification of nucleotide sequence present in this wonder molecule, that is the DNA. About 99.9% of the nucleotide sequences in all persons is the same. Only some short sequences of nucleotides differ from person to person. In the population, every person shows unusual sequences of 20 to 100 base pairs which are repeated several times. They are termed as variable numbers of tandem repeats, VNTRS. The length of the regions having VNTRS is different in each individual and hence is the key factor in DNA profiling. Have a look at this image here. There are 4 participants, participant 1, 2, 3 and 4. And have a look at the repeat in the sequence, the GATA sequence. Here you will, if you count 1, 2, 3, 4, 5, 6, 7, 8, it's repeated 8 times. In this 2 participants, the same sequence is repeated 9 times. In the 4th participant, the same sequence is repeated 10 times. So these students are called as short tandem repeats, which are also called as STRs. So in our DNA profiling, we have 2 types of these sequences, that is variable number of tandem repeats, VNTRs. The number of repeats vary from person to person. It's just 9 or maybe to 80 bases in length. And the short tandem repeats, which you just saw, is just 2 or 5 bases in length. Now, both these are used for analyzing data, tissue matchings, and also to understand inheritance patterns. Students, now let us have a look at the steps that are involved in DNA fingerprinting. The first step is isolation of the DNA. The DNA must be recovered from the cells or the tissues of the body, that is host. Only small amount of tissue like blood, hair roots, skin, etc. is required. The next step is restriction digestion. So you have your tissue sample and then the next step we move to restriction digestion. The isolated DNA is treated with restriction enzymes. The restriction enzymes cuts the DNA into small fragments having variable lengths. This phenomenon is called as restriction fragment length polymorphism, RFLP. So here is your DNA. It is then taken out and then it is subjected to restriction digestion. So the process moves forward and small fragments of this DNA are prepared. Students, the third step is gel electrophoresis. Now, the DNA samples are loaded from agarose gel electrophoresis under an electric influence. So this is the gel agarose gel which is going to be subjected for the process called as electrophoresis. Now for this, the DNA fragments which are negatively charged, they move to the positive pole. So once the DNA is loaded here, the negatively charged DNA moves towards the positive pole. Now the moment of these fragments also depends on the length of the fragments. Now this results in the formation of band, and the double-stranded DNA splits into single-stranded DNA by alkali treatment. Students, the fourth step is Southern blotting. The separated DNA fragments are transferred to a nylon membrane or a nitrocellulose filter paper by placing it over the gel and soaking them with the filter paper overnight. Here, have a look at the step in this figure. You will see that the membrane is placed on a nitrocellulose filter paper is placed on the agarose gel, and it is allowed to get blotted or get soaked in the filter paper. The next step is selection of the DNA probe. A known sequence of single-stranded DNA is prepared. It is called as a DNA probe. DNA probe is obtained from organisms or prepared by complementary DNA preparation methods. The DNA probe is labeled with radioactive isotopes. Students, after selection of the DNA probe, the next important step is hybridization. Probe DNA is added to the nitrocellulose filter paper containing the host DNA. The single-stranded DNA probe pairs with the complementary base sequence of the host DNA strand. Have a look at this tray here. So the membrane that is there, it is flooded with the single-stranded DNA probes. They form complementary base pairings. As a result, DNA-DNA hybrids are formed on the nitrocellulose filter paper. Remaining single-stranded DNA probe fragments are washed off. Then the final step is the photography. The nitrocellulose filter paper is photographed on an X-ray film by autoradiography. The film is analyzed to determine the presence of the hybrid DNA. So these DNA probes, as you know, are radioactive. So on an X-ray plate, the places where they bend, they show such an image where you have the autoradiography is done and the bands are observed. Students, let us have a look at the application of the DNA fingerprinting. The first is, in forensic science, DNA fingerprinting is used to solve problems of rape and some complicated murder cases. Now, how this is done? See, the entire process remains the same. You take the sample, then the step is to take the DNA, digest it with the restriction enzymes. Then the fragments are planted on for the process of electrophoresis. The bands separate depending upon their lengths. Then they are absorbed on a membrane. That is, they are transferred on a membrane, and then that membrane is flooded with the radiolabel probes, and then the image is taken. An X-ray image is done. Now, here how this helps in forensic is, you take the blood sample that was obtained in the crime scene, and then you can take the blood samples of the suspects. Now, here you will see there is an image of the crime scene, and you have the image of suspects 1, 2, and 3. And if you have a closer look, you will see that the image of the suspect 2 matches with the crime scene. So this is how the criminal is identified. Now, how this is possible? Can you see here? Have a look at this image now. You can see here these chromosomes 7 to 16 are shown here, and the short tandem repeats are shown here. So this is the blood sample from the crime scene, and this is how it is going to see on your X-ray film. And then you have 2 suspected individuals. So these are the tandem repeats of individual 1 and individual 2. Even if you compare the tandem repeats here, you will see that the crime scene tandem repeats matches with that of the individual B, or the second individual. And now once the entire process is run and the DNA fingerprint is generated, you will see that the sample C, that is the crime scene sample, matches with the individual B, or the second individual. So this is how it helps in identifying criminals. Of DNA fingerprinting is, it is used to find out the biological father or mother or both of the child in cases of disputed paternage. So if you see here, you have the mother's banding, the father's banding, and these are the children banding. So it is used to identify or eliminate, suspect, determine paternity of a child, or establish other family relationships, identifying a decreased person. So here is another application of it. So it is also helped in understanding inheritance pattern of a particular gene in a family. So you have the parents, you have the siblings, and then you can identify their genotype, whether they are homozygous or heterozygous for a particular trait. The third application of DNA fingerprinting is it is used in pedigree analysis in cattle, dogs, horses, and humans. So pedigree analysis is the study of transfer or inheritance of genes in a particular family. So this is how it is used to analyze the species or the breed of dogs, the breed of cats also. It's used in the pedigree analysis of horses. Here you can see a chart. This is a chart to trace a particular horse's breed. This is a very important process in a very famous sports where you have the horse riding that is done, the horse racing. And here it is very important to know the breed of the horse, that is whether it has the genes to run fast. So this is how the pedigree analysis of the horses are done. Then you have a separate category where your DNA fingerprinting is used, that is your human pedigree analysis. Now pedigree analysis in humans is to identify the inheritance of a particular gene in your entire family lineage. Now these are certain symbols that are shown here which will help you to understand a pedigree chart. Like for example, the square represents the male, the circle represents the female. So when you have a horizontal line between the male and the female, that shows the matings. Then you have the vertical line downwards which show you the offsprings. Then you have the shaded squares and the circles which tell you that it is the affected individual. If it is half shaded and half plain, then it is heterozygous for a particular trait. Then you have a circle with a dot that represents a carrier female. So all these symbols are used to show you the inheritance of a particular gene in a family lineage. For example, have a look at this chart here. The key is given, you have the male and the female. So the male is shaded, so the male is colorblind, and the female has the normal vision. So the female is not shaded here. And then when you move to a downwards line after the mating, you have them showing their offsprings, and the two offsprings are two daughters, and when they marry colorblind male, then their further offsprings move downwards. You will see in the lineage the females are not colorblind, but the male is colorblind. So this is how a pedigree chart of humans is drawn for analysis of a inheritance of a particular trait. Thank you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe, and keep learning. With this, we come to an end to this chapter of molecular basis of inheritance.